I am again delighted to bring to you this class today, and I want us to look at these questions. The instructions are very clear, and they're saying answer all questions in the spaces provided after each question. The first question given to us states, study the illustration below and answer the questions that follow. And of course, if you look at this diagram, you will realize that we have three main types of blood vessels. We have blood vessel A, we have blood vessel B, and blood vessel C. And of course, you have what you call the stomach and the intestine. The organ given here is the liver. And of course, you've actually been given that the information provided here states that the blood vessel C is carrying blood from the stomach and intestine and to the liver. So the first thing you're supposed to understand is this. The largest organ is always the skin. So for your information, the largest organ in the human body is the skin. After the skin, the second largest organ in the human body is the liver. For your information, the liver is the second largest organ in the human body. For your information, you have been told about the skin covering almost each and every part of your body. And of course, it has two major layers. You have what you call the epidermis as the upper layer, and the dermis that just lies below the epidermis. So you can be able to have the dermis and the epidermis. Those are the two major layers of your skin, the epidermis and the dermis. Very good. So the epidermis and the dermis. I've told you the outermost, or the one that is actually to the outside, is called epidermis, and the one below the epidermis is dermis. So dermis lies just below the epidermis. So, and of course, you have to look at other parts of the skin which can be able to involve the subcutaneous layer. We have what you call the sebaceous gland, you have what you call the rectal pili muscles. We can be able to have what you call Pacinian corpuscle. We can be able to have what you call the blood vessels, the nerve endings, the sweat glands, the sweat duct, and of course, all those other parts of the skin. And of course, if you talk about the liver, the major parts of the liver can be able to get referred to as the following. Number one, we have what you call the hepatocytes. So this liver is majorly made up of cells called hepatocytes. So if someone asks you to give the name of the cells making up the liver, you'll talk about the hepatocytes. And of course, hepatocytes are the cells of the liver that produce bile juice. Bile juice. Then the bile juice is directed into what you call the gallbladder, where it gets stored temporarily. And the bile juice will leave the hepatocytes as the cells making up the liver to the gallbladder through channels called canaliculi. So the channels allowing the bile juice to find its way into the gallbladder are called canaliculi. So I've said the liver is the second largest organ. It is made up of cells called hepatocytes. This is where bile juice is produced and as well secreted to the channels called canaliculum that direct this bile juice to the gallbladder, where this bile juice is temporarily stored, awaiting food to arrive. And once the food reaches in the duodenum, the bile juice is released by the gallbladder through what you call the bile duct, and this bile juice will act on the fats, breaking them down into more soluble forms that can easily get absorbed into the bloodstream. So you see, we are supposed to understand that this bile juice is just made up of what you call salts. So bile juice is just made up of bile salts. We have quite a good number of salts that you can be able to mention, but again, at this level, you can only be able to refer to two very important salts. We have what you call sodium taurocholate, and sodium glycocholate. So those are the two main types of salts that can be able to make up the bile juice. And these sodium taurocholate and sodium glycocholate salts making up the bile juice are very, very, very important in breaking down large fats into tiny fats. And the process of breaking down large fats into tiny fats in biology is referred to as emulsification. Emulsification. So the work of sodium taurocholate and sodium glycocholate as bile salts making up the bile juice is to ensure that fats are broken down from their large forms into the small or tiny forms. And that process is referred to as emulsification. And away from that, you can be able to come back and understand this. We said that the introduction of food into the mouth is called ingestion. And of course, you have been told about the salivary glands, which are salivary amylase producing glands. That's why we are actually referring to them as salivary producing glands. That means there are three. They produce saliva. We have what we call sodium oriented producing what? Glands. Because as part of what you have in terms of this saliva, we are going to understand that there is what you call parotid gland, the submandibular gland, and the sublingual gland. We are told that the parotid gland is found in the cheeks and just close to the ears, that is. The submandibular gland is found under the jaws, of course, what you call the mandible. Of course, you have to actually understand that human beings have two major parts making up what you call their face, or what you call the jaws. Of course, we have what you call the upper jaw and the lower jaw. So the upper jaw is made up of a bony part called the maxillae, and the lower jaw is made up of a bony structure referred to as the mandible. So the submandibular gland producing saliva, and may contain so many things, including even sodium salt for that matter, will always be able to get found just below the mandible or just below the jaws. And that means you can be able to talk about the sublingual gland that is found under the tongue. Those three glands will produce saliva. Saliva will be able to contain mucus that can be able to lubricate food and lubricate the lining of the mouth to prevent it from getting digested by the salivary amylase enzyme found in the saliva. At the same time, we have what we have as water, which can be able to soften food, be able to dissolve the food, be able to provide a very conducive environment in which the breakdown of food can occur, more specifically starch, because the breakdown of starch starts in the mouth, where the starch is broken down to a very important product called maltose. So salivary amylase, also referred to as ptyalin. So it's called salivary amylase or ptyalin, as the first enzyme to mention in our digestion process, is very important in breaking down starch to maltose. So that translates into something else. What you're supposed to understand is this. This salivary amylase or ptyalin breaks down a very important component of our diet called starch into what you call maltose. And of course, the tongue will roll the food into small round balls called boluses, and the tongue will still push the food at the back of the mouth. That means absorption has actually to start now getting near with the time. They are saying that now, when the food gets swallowed, it will end up actually passing through what you call the esophagus or the gullet. The longitudinal and the circular muscles contract and relax, and in the process, the food gets pushed down the gullet or the esophagus. The food enters the stomach through an opening called the cardiac sphincter, and of course, the cardiac sphincter allows room for the food to enter the stomach. The stomach has a lining called the longitudinal and the circular lining, of course, getting made up of those two very important muscles. Those muscles as well contract and relax, mixing the contents of the stomach. And in the process, there's a triggering effect on what we call the gastric gland in the glandular layer of the stomach, which then produces a very important juice called gastric juice by the effect produced by the gastrin hormone. And that ends up producing a juice that contains hydrochloric acid, which can be able to kill germs and provide a very acidic environment for the working of the enzymes in the stomach. The mucus will lubricate or act as a barrier, lining the stomach lining to ensure that the stomach is not corroded by the acid and as well not digested by these enzymes. Remember, in the mouth, the mucus lines the lining of the mouth to prevent the lining of the mouth from getting digested by ptyalin or salivary amylase. And you should also understand that at the end of the day, the food gets broken down. What happens here? There's an enzyme called pepsinogen, which gets activated to pepsin when the acidic environment is very conducive, brought about by the hydrochloric acid, which is a very strong acid in chemistry. That environment converting pepsinogen to pepsin makes pepsin to break down proteins to peptides. The digestion of proteins begins in the stomach. And as well, we can be able to have an aspect of this food getting pushed now out of the stomach through what you call the pyloric sphincter, a ring of muscles just at the exit of the stomach. The food reaches, of course, in the duodenum, the upper part of the small intestine. Of course, the small intestine has three major parts. One, the duodenum, the upper part. We have what we call the jejunum, the one in the middle, and of course the ileum, the lower part and the longest. So duodenum, the upper, jejunum, the middle part of the small intestine, and the ileum, the lower part of the small intestine, where digestion ends and is where food gets absorbed into the bloodstream. And you see, at the end of the day, in the duodenum, there are two very important secretions getting brought into this area. Number one, we have what we call the pancreatic juice from the pancreas and what you have as the bile juice from the liver. These two juices are able to get emptied into the duodenum via the bile duct and the pancreatic duct, respectively, from the liver and the pancreas. These two juices getting produced alongside a very important salt called sodium hydrogen carbonate. And of course, all these juices at one point must contain salts, like sodium salts, including the saliva in the mouth. And these juices, alongside the sodium hydrogen carbonate getting produced into the duodenum, is under the influence of two very important hormones. The first hormone is called sodium oriented producing or secreting hormone. These hormones, let me just mention them. One, we have what we call the cholecystokinin hormone. You must have heard of it, cholecystokinin. And we have what we call secretin, cholecystokinin and secretin. The pancreatic juice contains trypsinogen. The alkaline environment of the duodenum converts or actually stimulates or actually activates trypsinogen to trypsin, which then breaks down any remaining proteins into peptides. At the same time, you'll realize that in the pancreatic juice, there's what we call the pancreatic amylase, breaking down any remaining starch to maltose. Because the starch breakdown began in the mouth, and not all the starch can end up getting broken down. So any remaining starch can get broken down to maltose in the pancreatic aspect in terms of the pancreatic amylase. And of course, we have said the breakdown of proteins begins in the stomach by the aid of the pepsin enzyme. So any remaining proteins not broken down to peptides can get worked on by the trypsin enzyme found in the duodenum. Now at that point where it actually has to act on the remaining proteins. You have to talk about sodium hydrogen carbonate. The work of the sodium hydrogen carbonate is to ensure that an alkaline environment is provided in the duodenum for the effective functioning of these enzymes. And you see, at the same time, this same sodium hydrogen carbonate acting as an alkaline oriented component will always be able to ensure that the acidic food from the stomach is neutralized, because in the stomach the environment is very acidic. So any food leaving the stomach under any acidic condition can bring a disadvantage to the process of digestion in the duodenum. So the sodium hydrogen carbonate will ensure that all the acidic food called the chyme, that is from the stomach, gets neutralized so that an alkaline environment is provided in the duodenum. We have what we call bile juice. One, I've told you it contains two salts, sodium taurocholate and sodium glycocholate, to act on proteins, no, to act on large fats, breaking them down into tiny fats through a process called emulsification. The same bile juice provides a very alkaline environment for the working of the enzymes in the duodenum, and as well will also contribute to the neutralization of the acidic food called chyme from the stomach. These components are then taken into the ileum, the lower part of the small intestine. In the ileum, there is an enzyme oriented aspect of digestion of food, what you call chemical digestion, where when the food digests in the ileum, succus entericus or intestinal juice gets produced. This juice will be able to contain several components. What are these components? We have what we call, of course, crisps of Leberkuchen, as part of what we call the ileum, will be able to produce this juice. The peptidase breaks down now peptides to amino acids, the end product of digestion of proteins. On top of that one, you'll realize that the maltase breaks down maltose to glucose, the end product of digestion of starch. Sucrose breaks down sucrose to glucose, the end product, of course, glucose and fructose. the end products of digestion of starch. And of course, you'll also have lactase, you'll also have lipase. All these end the process of digestion. For example, lipase can break down uh, any lipids, any fats, or any oils into fatty acids and glycerol. Look at that. And remember, in the duodenum, there is an enzyme called pancreatic lipase, which breaks down lipids into fatty acids and glycerol. In that process, any remaining lipids can get broken down in the ileum into fatty acids and glycerol. And of course, all this, like lactase, breaks down lactose to galactose and glucose. So they have actually to end up ending the process of digestion of food in the alimentary canal or along the gut. As I proceed, students, you'll realize that digestion has ended. The ileum is well modified to actually ensure that digestion has actually ended and all the nutrients ending from this process are able to get absorbed into the bloodstream. Of course, you have what you call glucose, amino acids, fatty acids, and glycerol. Of course, you have what you call water, some mineral salts, some vitamins, and of course, the better part of absorption of water into the bloodstream occurs in the large intestinal colon. But the absorption of glucose amino acids will always be able to occur in the ileum alongside fatty acids and glycerol. For example, glucose and amino acids can get taken into the bloodstream through either diffusion or active transport. And fatty acids are first emptied into what we call lacteals. And the lacteals can now be able to actually empty these contents into what we call the lymphatic system, which will later join the normal circulatory system through what we call the subclavian vein. So I think after digestion has ended, nutrients have been absorbed into the bloodstream is when now they can get transported to the liver through what we call blood vessel C. And blood vessel C is a hepatic portal vein. It takes all the nutrients of digestion from the intestines, of course the stomach, and into the liver, where an excess of these nutrients can get converted into forms that can get stored. For example, the excess of fatty acids and glycerol, for that matter, the excess of glucose, the excess of amino acids can be a problem to the body of a human being. Remember, human beings don't have a mechanism of storing excess proteins, and as well, any excess glucose in the bloodstream can translate into diabetes mellitus. So there must be a mechanism of actually controlling these amounts in the bloodstream, because if this blood is left to circulate and move around the body with this excess products of digestion, it can be a challenge to the body in terms of surviving. So for example, in the liver, when the blood reaches here, any excess glucose can get converted to glycogen by a hormone called insulin. Insulin is secreted by the pancreas. Remember, in the pancreas, we have what we call the beta cells, the alpha cells, what we call the islets of Langerhans. So remember, islets of Langerhans as cells found in the pancreas produce what you call insulin, which can now be able to convert any excess glucose to glycogen. The glycogen gets stored in the liver and of course in the muscles for that matter. Any drop in the level of glucose in the body can actually trigger the pancreas to produce another hormone called glucagon, which can now convert the stored glycogen back to glucose to raise the level of glucose in the bloodstream. So those two hormones are very antagonistic, working against each other in the opposite direction. And of course, the liver is the second largest organ, receiving a large amount of blood from the normal circulation of a human body. Which blood vessel brings the blood to the liver? Of course, maybe from the heart, when there's what we call the sub-branching, when the outer branches. That blood vessel is called hepatic artery. Of course, arteries carry blood away from the heart, and the blood is oxygenated. Apart from two major arteries which carry deoxygenated blood, that is pulmonary artery and another artery called umbilical artery, which carries deoxygenated blood from the fetus to the mother's circulatory system. And of course, pulmonary artery carries deoxygenated blood from the heart to the lungs so that the blood can get oxygenated. And of course, the blood that leaves the liver through blood vessel A is called hepatic vein, carrying blood towards the heart, mostly deoxygenated, except two blood vessels. Now, the two blood vessels carrying deoxygenated blood, in as far as veins are concerned, are number one, we have what we call the pulmonary vein, carrying blood which is oxygenated from the lungs back to the heart, and the umbilical vein carrying oxygenated blood from the mother's circulatory system to the fetus. to supply nutrients and oxygen, and of course antibodies for that matter. So of course you can be able to answer these questions. The blood vessel A is called hepatic vein, blood vessel B is called hepatic artery, blood vessel C is called hepatic portal vein. Wow. So this is a hepatic vein. This is called hepatic artery. This is called hepatic portal vein. And there you go with 3 marks in your pocket. So give one difference in composition of blood found in A and B. So in composition, in other words, what does this blood contain, or what is it made up of in terms of this blood vessel A, and this blood found in blood vessel B contains which components? One thing to note is this. Number one, the blood leaving the liver is able to contain a large amount of nutrients. Why? The blood has just found itself through this blood vessel C. This blood contains end products of digestion. When the blood reaches in the liver, it contains nutrients like glucose, amino acids, fatty acids, and glycerol for that matter in small quantities, vitamins, mineral salts, and other components. And of course, at the end of the day, when this blood vessel leaves the liver, it contains a controlled amount, lower concentrations of different components of the end product of digestion. This one is just leaving the heart and coming back to the liver with blood which is oxygenated, lacking the end products of digestion. So this blood contains end products of digestion, such as glucose and amino acids. This blood doesn't contain end products of digestion, such as glucose and amino acids. Remember, when this blood leaves, it has a lower concentration of oxygen. Why? This is the hepatic vein getting back to the heart. It is deoxygenated blood, so it contains a very low concentration of oxygen. This one is actually leaving in the heart, coming back to the liver with blood which is actually oxygenated. So the blood in blood vessel B contains a large amount of what? Oxygen. Very good. So that question is well answered. Blood vessel A contains end product of digestion, such as glucose and amino acids. Blood vessel B does not contain end products of digestion. Blood vessel A as well have said is deoxygenated, containing a very low concentration of oxygen. Blood vessel B is oxygenated, containing a large amount of oxygen in its blood in terms of what it's carrying. Let's go to the question next to this. My phone has a problem. So what characteristics of living organisms is shown by millipede coiling when touched? It is irritability, or you can also be able to talk about sensitivity. Irritability or sensitivity. An athlete sweating. Of course, sweating is a way in which excess water, some mineral salts, traces of urea can get removed from the body through the pores on your skin. So when someone does the sweating or actually undergoes sweating, then that means that is a form of excreting the wastes from the body. One, excess water. Don't talk about water, excess water. Water is a very important component of our body. In fact, 70% of our bodies are actually made up of what? Water. So then that means we are actually excreting. So the process or the characteristic is excretion. That means excess water, some mineral salts, and of course when you test your sweat, it's actually salty. That means some salts can get found in the sweat. So excess water, some mineral salts, and traces of urea can be able to form sweat, and that is a way in which you can be able to excrete waste from our body through the skin. So skin is an excretory organ, apart from the liver, and of course the kidney, the lungs, and other parts of the human body. Yeah, and of course someone would have actually thought of something like respiration, which means maybe the chemical breakdown of food in the presence of oxygen in the cells of living organisms to produce energy. So, and I think at the end of the day, the energy produced in form of heat, and heat translates into the formation of sweat. So, but a bit closer to this description is excretion. Wow, so I think you can stop there and then you can proceed to the next part of our question in our next lesson.